while we still have a lot of non-vascular plants, plants have evolved past this attachment to water and being in moist habitats, thanks to the next great leap forward in evolutionary terms, which is vascular tissue. So, what's so great about having vascular tissue? Well, perhaps something that's not great about having vascular tissue, but a repercussion of having vascular tissue is the sporophyte generation becomes dominant. So remember, in the non-vascular plants, the gametophyte was the dominant generation, and the sporophyte was usually dependent on the gametophyte for its nutrition. But with vascular plants, all the rest of the plants we're going to talk about, the sporophyte generation has become dominant. The gametophytes in vascular plants are very variable. In some cases, they don't photosynthesize. In some cases, they might be attached to the sporophyte or not. The gametophytes may be found above ground or they may be found below ground. In our seedless vascular plants, the gametophytes we don't often see. So for this reason, seedless vascular plants have historically been called vascular cryptogams, meaning the gametophytes are cryptic. We don't see them. But by and large, vascular plants are larger than non-vascular plants. This enables them to move water into their aerial tissues and inhabit drier habitats, which gives them the advantage of being able to have better access to sunlight so that they can photosynthesize and outcompete the smaller non-vascular plants. So being a vascular plant means that you are comprised of a series of tubes, but you also can develop tissue systems and complex organs. There are three fundamental tissue systems: the dermal tissue system, the vascular tissue system, and the ground tissue system. So the dermal tissue system provides an outer layer, kind of analogous to our skin, that helps keep moisture inside the plant and protect the plant from predators and pathogens and other things that might damage it. Provides a little bit of strength to the outside. The vascular tissue system is comprised of these tubes of xylem and phloem. Xylem is the vascular system that takes water and mineral nutrients from the soil and the roots up into the aerial tissues, the stems and the leaves. Phloem is the tissue system that moves photosynthate, or fixed carbon or sugar, from the places where it's made, the photosynthetic tissues, to wherever it's needed. Phloem also moves hormones around. If a plant needs to heal over a wound, phloem will be the system that's going to move those resources that are needed to the site of the wound, for example. So xylem moves pretty much unidirectionally from soil up into the air, and phloem will move resources wherever they're needed, so sideways, up, down, wherever it's needed. The ground tissue system, this is ground in the sense kind of like it's in the background. It is not dermal and not vascular, so it's kind of filling space between the dermal tissue system and the vascular tissue system. It's not ground as in soil, it's not root tissue, although roots have ground tissue in them. And the ground tissue system can be used for storage. It can be used to provide structure and strength to the stems of vascular plants. So let's take a look at some of these tissue systems. All right, so In this diagram, we've got two types of flowering plants, or very advanced vascular plants. Here's a monocot, here's a dicot, and the outer dermal layer is represented in blue. So we've got epidermis, a few cell layers thick. We've got the ground tissue in yellow. You can see this makes up an awful lot, the majority of the tissue inside the dermal layer, and then the vascular tissue is a relatively small component, but very important component of the plant stem. So in addition to having these tissue systems, we also get organs, true organs, roots and stems and leaves. So in the non-vascular plants, mosses don't have roots, they have rhizoids. They don't have true stems and they don't have true leaves. In order to have those structures, you have to have vascular tissue present. As far as the leaves go, there are a couple of different ways that a plant can make a leaf. Any given type of plant will produce one of these types of leaves. So the most common are megaphylls, but within the Lycophyta, we only see microphylls, also Equisitophyta. What makes a microphyll, the name means small leaf, but what's really happening is that we've only got one strand of vascular tissue in each leaf, which makes leaves tend to be very fine, very thin, but they can still be very long. Megaphylls typically have multiple strands of vascular tissue in each leaf. So here you can see, for example, here's a member of Lycophyta, Selaginella kraussiana, which has microphylls, only one strand of vascular tissue. And ferns, for example, have megaphylls where you've got branched vascular tissue. So the significance of this is reflected in the evolutionary history of these plants. So we've got two lineages of seedless vascular plants that have microphylls, and they are very different from these lineages that have megaphylls. And it's thought that megaphylls evolved through branching of the vascular tissue and the development of blade material that expanded to produce these very broad megaphyll leaves. In terms of growth and development, since we have these tissue systems, we can also have different types of meristems. And meristems are, in plants, zones of active cell division and growth. Kind of like stem cells in animals, meristems are cells that can divide and become other types of cells. So apical meristems are found at the apices of the plant, so at the end of the root and at the end of the shoot. These are responsible for primary growth or elongation. You can increase a plant's height only using an apical meristem. But of course, plants are growing in both height and in depth because the roots, almost half of a plant's biomass, in many cases more than half of a plant's biomass, is found underground. Lateral meristems are also present, or axillary meristems they are sometimes called. These are responsible for secondary growth, or an increase in girth. So the stem of a tree, you may know, develops a new annual ring every year, and that's the product of secondary growth produced by lateral meristems rather than the primary growth of the apical meristems. We'll talk more about secondary growth at the end of this unit on plants. But here is a diagram showing how we have terminal buds that have apical meristems. We also have an apical bud at the root apex. Last semester, Whenever you may have taken Biology 1020, you may remember taking garlic bulbs and removing the rootlets to look for cells going through mitosis. And you were told, look just in the first few millimeters of your rootlet, because you're not going to see active mitosis much further beyond that, because you're looking at the root apical meristem of that garlic bulb. And the lateral meristems, or axillary meristems, are found at the base of leaves as they produce new branches. Also, in vascular plants, we see a shift from strict homospory, or having only one type of spore, to heterospory, or having more than one type of spore. And by more than one, I mean two types of spores. They are large spores and small spores, or microspores and megaspores. Micro are the small ones, mega are the big ones. So we've historically considered 4 phyla of seedless vascular plants: Pteridophyta, Equisitophyta, Psilophyta, and Lycophyta. Some of these phyla have been given different names for the same taxonomic grouping, but it's been found that 3 of these phyla form a clade, which our text refers to as the Monilophyta, which I will consider as a superphylum consisting of the phyla Pteridophyta, Equisitophyta, and Psilophyta. But Lycophyta is definitely something different. It's a different phylum, has a different evolutionary history relative to these other 3. So here is a phylogeny, or evolutionary history, of the kingdom of plants, starting with our ancestral charophyte green alga ancestor. The first land plants were things like mosses and liverworts and hornworts, the non-vascular plants. And then when we get vascular tissue become sporophyte dominant, we see things like the Lycophyta or lycophytes, and then the different types of ferns, whisk ferns and horsetails in the Monilophyta. And then later on, we'll talk about what happens when you develop seeds. So let's talk about the first phylum, the phylum Psilophyta. It's only first in that I'm talking about it first because it's fairly simple. Now, it's simple not based on evolutionary advances in other plants. This Psilophyta has been found has actually lost a lot of these types of structures like true leaves and roots. We identify whisk ferns, or members of Psilophyta, based on their dichotomously branching stems and the sporangia that are borne directly on the stems. So here is an example of Psilotum. You can see the dichotomously branching pattern. So every time there's a fork, you can see the 2 limbs of that fork are more or less equal in size. So that's what we mean by dichotomously branching. So every time you see a branch point, you've got 2 equal lobes, or bifurcations off there. And all these little green dots on there, those are the synangia, which is the specific name for the spore-bearing containers where meiosis occurs in Psilotum. Here's a close-up of one of those sporangia. You can see it's got 3 parts, and it splits open and releases the